In biology, some of the absolute biggest discoveries, well, they come from the smallest places, a world that's completely hidden from our view. So how do we actually see it? Today, we're going to crack open the toolbox that makes it all possible and take a journey into that unseen microscopic world. I mean, that's the big question, right? Think about it. From the teeniest bacteria to the incredibly complex cells that make you, well, you, there's this entire universe that we just can't see. So where do we even begin? Our journey starts with the simplest tool of them all. All right, we're starting with the very first tool in our kit, the hand lens. Yeah, before we get to the super high-powered scopes, we start right here. Think of it like a detective's magnifying glass. For a biologist, it's that very first peek into a much, much larger world. And the design is just brilliantly simple. I mean, at its heart, a hand lens is just a convex lens that's a piece of glass that's curved outwards to bend light, and it's stuck in a frame with a handle. That's it! It's made to be easy to hold, so you can get that first, quick, closer look. So this brings us to a super important idea, magnification. It's not magic, it's actually pretty straightforward. It's just a way of measuring how much bigger something appears compared to its actual size. And really, that's the whole point of all these tools, to make things bigger. And yeah, we can totally measure it. The formula is really simple. You just take the size of the image you see and divide it by the size of the real object. So let's say a bug is 1 centimeter long, but through your lens, it looks like it's 5 centimeters long. Well, you've got 5x magnification. See? Simple as that. But a hand lens can only get you so far, you know? To really, truly see the secrets of the cellular universe, we need a huge leap in power. So it's time to meet a biologist's best friend, the compound light microscope. And here it is, this beautiful machine. It literally changed biology forever. Now, it's called a compound light microscope for two really good reasons. First, it uses a beam of light to light up whatever you're looking at. And second, it's compound because it uses more than one lens to magnify the image. That's what gives it some serious power. I know, it might look a little complicated with all those knobs and lenses, but trust me, we can figure this out pretty easily by breaking it down. We're going to take a quick tour, and we'll group all the parts by what they do. Think of it like 4 systems all working together in perfect harmony. First up, let's talk about the structure. This is basically the skeleton of the microscope. You've got the base, which keeps it steady on the table. The arm is what you use to carry it. And the stage, that's the flat platform where you put your slide. These parts are the solid foundation for everything else. Okay, next is the light system, or the journey the light takes. It all starts at the bottom with a mirror, which bounces light up. That light then goes through the diaphragm, which is kind of like the pupil of your eye, controlling how much light gets in, and then through the condenser, which focuses all that light right onto your specimen. And now, for the magic, the vision system. This is where the real work happens. You've got the eyepiece, that's the lens you actually look through. And then you have the objective lenses, which are on that rotating turret. Those are the ones right above the object, and you can switch between them for different levels of power. And finally, the focusing system. This is how you get that perfectly sharp, crystal clear image. The big knob, the coarse adjustment, moves the stage up and down really fast for a quick, rough focus. Then the little knob, the fine adjustment, makes teeny tiny movements to really dial it in. It's like going from a rough sketch to super fine details. So with 2 different lenses working together, the eyepiece and the objective, how powerful is this thing really? Let's figure out how to calculate its true total power. This is a skill you're going to use all the time in the lab for sure. So here's the secret. The total magnification isn't just one lens. It's the power of both of them combined. And the formula? It could not be easier. You just multiply the power of the eyepiece lens by the power of whatever objective lens you're currently using. So let's run the numbers. Say your eyepiece has a pretty standard power of 10x, and you click the high power objective lens into place, and that one's 40x. What do? You multiply. 10 times 40 is 400. That means what you're seeing is 400 times bigger than it is in real life. That is some serious power. Now, an instrument that's this powerful is, you guessed it, also really delicate and pretty expensive. So being a good biologist means you gotta know how to handle it like a pro. There are just a few simple, but totally non-negotiable rules for taking care of your scope. All right, first rule, always carry it with 2 hands, one on the arm and one supporting the base, always. Second, keep it upright. You don't want the eyepiece to just fall out. Third, this one's a bit obvious, but yeah, never ever drop it. Fourth, only use that special soft lens paper to clean the glass. Anything else will scratch it for good. And last, when you're all done, put it away safely in its box. Treat it right and it'll show you incredible things. And this right here, this is why we do it. On the left, you see a single-celled bacterium, and on the right, you can see the complex, specialized cells in a plant root. Two totally different forms of life, both completely invisible to us, but both brought into sharp focus by the microscope. This tool shows us the fundamental building blocks of everything. So why does learning this stuff matter so much? Because knowing your way around a microscope isn't just for biology class. It's the foundation for some amazing careers. Seriously, think about it. Every doctor, every pharmacist, every scientist trying to cure diseases, they all started right here, learning how to use these exact tools to see how life really works. So, there you have it. You've gotten the full tour, from the simple little hand lens to the powerhouse microscope. You know the parts, you know the power, and you know how to handle them. The only question left is, now that you have the keys to an invisible kingdom, what unseen worlds are you going to discover first?